Good evening. My name is Hannah Antosh, and this is Glycolysis, Gluconeogenesis, and the role of Aldolase in gene expression. My name is Hannah Antosh, and this presentation is for Dr. Karma Cook for Chemistry 530, Concepts of Biochemistry. To get started, the goal of this presentation is to explore the glycolysis pathways, explore the gluconeogenesis pathways, how both of these pathways are regulated via aldolase, and to discuss how aldolase B expressions can be regulated by the transcription factors HNF1, HNF3, CEPB, and DPB. To get started, we're gonna begin with glycolysis. So glycolysis is a type of metabolic pathway that serves as a source of anaerobic energy and is present in nearly every organism. Almost every organism is able to undergo glycolysis in areas where oxygen may not be as widely available. Glycolysis is always the first step in cellular respiration. If we think back to any freshman biology class and learning about cellular respiration, we learned that it begins with the glycolytic pathway, in which an organism receives 2 ATP energy before going on to complete the full cellular respiration cycle and ending with like 32 ATP. So we see here, our fun little chart here shows us the breakdown of glucose into pyruvate, which we'll talk a little bit more about in just a second. So the glycolytic pathway, during the process of glycolysis, glucose is actually going to be broken down into pyruvate and ATP per that last chart. A total of 2 ATP energy will be at the end of this process. Glycolysis only occurs in the cytoplasm of the cell and can occur in both aerobic and anaerobic environments. In the aerobic environment, the pyruvate the pyruvate that's gathered from that initial glucose molecule will actually go into the mitochondria, and that will feed and trigger that oxidative phosphorylation process. In an anaerobic environment, that pyruvate stays in the cytoplasm and will convert back into lactate to begin enzyme lactate dehydrogenase. So we look back at this little chart here. Glucose, with the use of ATP, turns into glucose 6-phosphate, which is then broken down into fructose 6-phosphate. Which is broken down into fructose 1,6-biphosphate, which we can see aldolase right here, because aldolase is a very important enzyme in breaking down fructose, allows it to be broken down even further and further again. And we end up, through all of these steps, we end up with an end product of pyruvate and 2 ATP energy. Glycolysis in and of itself has 2 main phases. We have an investment phase and a payout phase. So in the investment phase, glycolysis begins when hexokinase and phosphorylating glucose can actually start breaking that glucose molecule down into a glucose 6-phosphate. This step is irreversible. This is commitment time for glycolysis. That phosphorylation captures that glucose molecule. Since it cannot easily pass back through the membrane, it's stuck right there. Phosphoglucose isomerizes G6P into a fructose 6-phosphate, which then, with the addition of phosphofructase kinase, adds a second phosphate back into the equation. Sorry, isomerase and isomerizes is a real tongue twister here. The next phase we come to is the payoff phase. Now, in that payoff phase, glyceraldehyde 3-phosphate dehydrogenase gets to turn that G3P into a 1,3-diphosphate glycerate through the reduction of NAD+ into NADH. Then that 1,3-diphosphoglycerate loses one of its many phosphate groups via phosphoglycerate kinase, which creates ATP by using a substrate level phosphorylation. At the end of the payoff phase, 2 ATP are produced. Remember, this is just a brief overview of glycolysis because it has too many phases. Now, glycolysis was when your body already has glucose to work with. Gluconeogenesis, however, occurs when your body does not have glucose readily available to it. You haven't eaten glucose, you haven't ingested anything with glucose. And so your body forms glucose from non-hexose precursors. It's another metabolic process that also has aldolase that influences it. So gluconeogenesis can occur from non-hexose precursors such as glycerol, lactate, pyruvate, propionate, and glucogenic amino acids. So gluconeogenesis is essentially reverse glycolysis. Remember, in glycolysis, we're breaking down from a glucose molecule into a pyruvate, and here we're going from a non-glucose molecule into a glucose molecule. There are 4 enzymes that support glucose synthesis by gluconeogenesis by reversing 3 exergonic glycolytic steps, specifically. Gluconeogenesis is actually triggered by low blood sugar levels. And it can also be triggered to clear lactic acid away from as like a waste product, because it can, remember that lactic acid can build up as a result of anaerobic respiration or from shock, which can make shock lethal. That's why shock can be deadly after like a major trauma or witnessing a horrific event. People can go into shock and that can be lethal. It partly due to the lactic acid buildup. Gluconeogenesis sounds great. It does, however, have a downside. It only produces a moderate supply of glucose. It cannot supply your whole body with all the glucose it needs every single day. This is kind of your body's emergency plan in a sense. This is not made to be your body's number one source of glucose. If the gluconeogenesis pathway happens to fail, most of the time it is fatal because your brain, eyes, and kidneys need glucose in order to function. Glucose is their main fuel source. So if they do not have glucose, they are not functioning. This means that glucose depletion is bad news, and that acute hypoglycemia can cause immense brain and kidney damage. I think I've mentioned before, my father was a type 2 diabetic, and he did not realize this intentionally or unintentionally, meaning that he didn't want to know he had type 2 diabetes until he went to the eye doctor because he was losing his eyesight due to his body's inability to break down glucose because of his insulin. Wild times. Now, gluconeogenesis is also quite immense in its steps, so we'll try to get through this a little bit quickly. So the first step of gluconeogenesis is that pyruvate carboxylase converts pyruvate into oxaloacetate. I think I said that right. To activate this step, to move pyruvate into oxaloacetate, ATP and biotin are required. This step occurs in the mitochondria. Now, after this step, the second step is that oxaloacetate is then converted into malate before it crosses the mitochondrial membrane. Now, once it crosses through that mitochondrial membrane, it's converted back into oxaloacetate once it reaches the cytoplasm. Remember that this is just the opposite of glycolysis. Remember that glycolysis happens in the cell membrane, and then pyruvate can go into the mitochondria. Here we're starting in the mitochondria and moving back into the cell membrane. Within the cytoplasm, phosphoenolpyruvate carboxykinase, also known as PEPCK, decarboxylates oxaloacetate. Now this is going to rearrange that oxaloacetate back into phosphoenolpyruvate, which is PEP. That PEPCK needs something called GTP and magnesium ions in order to activate. Enolase then moves in to hydrate those phosphenolpyruvates to create a 2-phosphoglycerate. Phosphoglycerate mutase turns that 2-phosphoglycerate into a 3-phosphoglycerate. Then we get something called phosphoglycerate kinase, then phosphorylizes that 3-phosphoglycerate in order to form 1,3-biphosphoglycerate. This step right here requires ATP energy in order to continue. Glyceraldehyde 3-phosphate dehydrogenase is then going to move in to reduce that 1,3-biphosphoglycerate into a glyceraldehyde 3-phosphate. BADH is an electron donor for this particular step of gluconeogenesis. Triose phosphate isomerizes the glyceraldehyde 3-phosphate to create dihydroxyacetone phosphate, also known as DHAP. Now, this is where our friend aldolase comes in. Aldolase comes in and combines with glyceraldehyde 3-phosphate, and DHAP in order to create fructose 1,6-biphosphate. Fructose 1,6-biphosphate then dephosphorylizes fructose 1,6-biphosphate into fructose 6-biphosphate. Now, this step is going to reverse that non-equilibrium reaction that was catalyzed originally by the glycolytic enzyme phosphofructose kinase 1. We're almost to the end here. Phosphohexose isomerase then turns that fructose 6-phosphate into glucose 6-phosphate. And then glucose 6-phosphate dephosphorylates glucose 6-phosphate into creating plain glucose. Now, glucose is able to enter into the bloodstream freely, and the body is able to use it immediately. Now, that's a lot of stuff. So remember that gluconeogenesis is just the opposite of glycolysis. Once you learn one, you learn the other, just backwards. Now, aldolase is a name that popped up in both of these scenarios. So aldolase, fructose 1,6-biphosphate aldolase, is a glycolytic enzyme that catalyzes the reversible breakdown of fructose 1,6-biphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Aldolase is a critical enzyme in breaking down fructose. If we didn't have it, we couldn't break down fructose, and that leads to a whole mess of trouble, as I said here. Now, specifically in gluconeogenesis, and that was specifically for glycolysis. Now, in gluconeogenesis, that aldolase B actually plays quite a significant role because of its role in breaking down fructose. Aldolase B specifically is a liver enzyme. Of the 3 aldolases, which we'll talk about later, aldolase B has the best catalytic efficiency rate for catalyzing both fructose 1 and fructose 1,6. This enzyme is so important during gluconeogenesis because it brings that glyceraldehyde and DHAP to create fructose 1,6-phosphate. That fructose 1,6-phosphate can then be catalyzed by the aldolase B liver enzymes. Now, like I said, aldolase has several different isoforms. There are 3 main ones. The first one is aldolase A. Now, aldolase A specifically causes the cleavage of that fructose 1,6-biphosphate into glycerol 3-phosphate and dihydroxyacetone phosphate. Researchers aren't entirely sure about aldolase's catalytic mechanism yet, so the full extent of the role that it plays is not available to us yet. We're still not entirely sure. The second one is aldolase B. Now, aldolase B is, again, just indispensable for fructose metabolism and gluconeogenesis. There is something that I referenced earlier called hereditary fructose intolerance. And this is a recessive trait, autosomal recessive trait, and it leads to elevated levels of fructose 1-phosphate. And this can be lethal if it is not, if it's not treated quickly enough. This spells bad news bears. Aldolase B also controls, the control expression of this enzyme, what controls that expression of aldolase B, is, again, not available to us yet. This is a great area of research. Aldolase B is expressed in the liver, kidney, and small intestines. The final form of aldolase is aldolase C. Now, this is located in the brain and other neurological tissues. It's probably the most enigmatic of the three aldolases. Its physiologic role is not quite all the way known yet. We think it might substitute for aldolase B in tissues when aldolase B might be lower than normal, but it's not all the way known. It's still enigmatic. It's still a mystery. So the three aldolase isoforms all have unique genes that are still related. So researchers think that they all came from one ancestral gene that throughout years of evolution has broken down into three separate genes with three separate jobs. In adult organisms, aldolase B is found in most of the body's tissues. It's generally concentrated It's found in most body tissues. Aldolase B resides in the liver, kidney, cortex, and small intestines, while aldolase C resides in the brain and nervous system tissues. Aldolase B specifically has some interesting regulation. There are four main regulatory genes that we're going to talk about, and the first one is CEPB. Now, CEPB is a very weak activator of aldolase B promoter. It can activate it, but it's not going to be the first one that you're, like, it's not the first one. It's fairly weak. And it tends to enhance distal negative elements. DPB, however, is a strong transactivator of aldolase B promoter. HNF1 is another strong activator of aldolase B promoter, while HNF3 is an extremely strong anti-activator of aldolase B. Now, just to recap, glycolysis occurs in the cell membrane, and it occurs when a pyruvate molecule is created by taking a glucose molecule and breaking it down and gaining 2 ATP plus a pyruvate. That pyruvate then moves in the mitochondria to begin aerobic respiration through cellular respiration. Remember that gluconeogenesis, however, is the exact opposite, meaning that it takes that pyruvate molecule, moves it out of the mitochondria through a couple different forms, and then breaks down from pyruvate back into glucose. Both of these metabolic pathways are influenced through aldolase because of aldolase's tendency, ability, job, role, however you want to say it, to impact the breakdown of fructose as a sugar. And remember that aldolase has three main forms: aldolase A, aldolase B, and aldolase C. Both aldolase A and C are a little bit more enigmatic. Their roles are not fully fleshed out yet. They're not fully known. While aldolase B is a little bit more fleshed out, we know that aldolase B occurs in the liver, kidney, and small intestines, while aldolase C occurs in the neuronal tissue, and aldolase A is in pretty much everywhere. Thank you so much. Have a wonderful night.